Okay, so my partner and I were splitting it. So I'm gonna attach all of my notes, which does include the variations in chromosome structure, deletion, terminal deletion, interstitial deletion, duplication, gene families, and globin gene family. So I split, like, that's what I'm not going to cover today. I'm going to let my partner cover that one. I'm going to cover mutations that change the location of genes. Okay, and then I'm going to cover inversion, the two different types of inversion, translocation, three different types of translocation, variations in chromosome numbers. There's four of those. Okay, so that's what I'm going to cover with this one. So mutations that change the location of genes are inversion and translocation. Inversion is when the segment has changed directions. So your segment might have your DNA segment might read certain numbers, or I'm sorry, letters, and then they get flipped and now they're read backwards. There's pericentric inversion, which is inversion of the central region of the chromosome and the centromere. This has the potential to change the position of the centromere. So the centromere, I believe, is when the chromosomes are in an X. Let me see. I want to look up. Oh, give me just a moment. I want to look something up here. So yeah, it's where they join together. So this is really vital to make sure that um, the cells are receiving um, the correct coding and the correct DNA. Um, so there's also paracentric. So that was paracentric, and this one's paracentric inversion. This is when the arm of the chromosome is broken and inverted, leaving the centromere outside the chromosome. When inversions alter the gene function, expression of the genes can result in cancer. So when inversions alter the gene function, overexpression. So maybe something that shouldn't be expressed is highly expressed. Okay. And this can also lead to cancer. So the inversion should not be on the outside, or the centromere should not be on the outside of the DNA of the chromosome. Again, I'm gonna look something up here. So let's see. So when the centromere is on the outside, the chromosome isn't able to um, function correctly. I wonder if maybe it means that some DNA could be lost um, because the centromere is on the outside. Then there's translocation. This is a piece of one chromosome transferred to another chromosome. So I think that we do see that when they cross. There is some, the DNA does switch, I believe, from one side to the other, or um, there are like the little arms and the longer arms. Um, I've got three different types of translocation. There's simple translocation. This is when a portion of a chromosome is attached to another chromosome, but the DNA remains together. Um, let's see. Let me see. So swapping genetic material between the two chromosomes. This is a good thing because it rearranges the DNA. It breaks it up. So that's good. Um, we need that. Then there's reciprocal translocation. This is when non-homologous, so DNA with different genetic makeup, chromosomes exchange portions. Chemicals and environmental agents can break chromosomes, which is not good. We should not have that. Um, this can form reactive ends combining non-homologous chromosomes. So the way I kind of understood that was, so non-homologous is the different genetic makeup. So they're not the same, right? And we need that. We need that variety. And we have chromosomes from our mother and chromosomes from our father, and they come together, and or DNA, and they come together in the chromosomes and they mix up. Okay. So from what I was understanding was these chromosomes can break, and when they break, they can then form together in ways that they shouldn't. And then we can get uh, like health issues. We could get what's the word I'm looking for? Let me see. Let me look here. Like impairments, like physical impairments, disabilities, I think. Um, there's also Robertsonian translocation. This is the function of long arms between two acrocentric chromosomes. I put on here that that means that uneven arms and the smaller portion is lost. So the arms, there's a long portion and a small portion and a long portion and a small portion. And when they cross, it looks like the smaller portions drop off and are lost. And that's really important because I believe the small portions contain a lot of DNA, like how to read that. And we really need that, or some of our DNA can be lost, causing other health issues, disabilities as well. An example of that would be familial Down syndrome. And typically the Robertsonian translocation is carried from the parent. One parent will have that translocation chromosome. So even if that doesn't come out in the parent, it can come out in the offspring. Then the last thing I'll talk about is variations in chromosomal numbers. So, that's variation in euploidy cells. I needed to look that up. I didn't know what that was. So that is a cell that has a normal number of chromosomes. Euploidy is a cell that has a normal number of chromosomes. I think that the normal number is 23, probably. So I have 4 different examples here of variations. There's the haploid, which is one set of chromosomes, which is totally normal. There's the sperm cells, and there's the egg cells. Then the next is diploid, which is the 2 chromosome sets, totally normal. One set from mom and one set from dad come together, and now there's you. Then there's triploid, which is 3 chromosome sets, and this is not normal. This is abnormal. An example of this would be seedless watermelons and bananas. Typically, chemicals are used to prevent chromosomes from splitting, which gives 3 sets of chromosomes instead of 4. So I think what that means is like there's a set here, and then there's another extra chromosome here, but it doesn't split, maybe. I think that that's what that means. Then the last one is the polyploid. This is more than 2 chromosomes. So my examples here are salamanders, frogs, and leeches. Salamanders, frogs, and leeches have a higher chance of gene expression with increased adaptive potential. So these creatures are able to be in an environment, and maybe these chromosomes are maybe they're causing gene expression that's not normal, but it's also not harmful. So maybe maybe adapting to being able to live underwater for a longer period of time, or coming out of the water for a longer period of time. There's also, we talked about how the frogs sometimes have a hard time with reproduction. And that would actually be helpful because then they wouldn't be able to reproduce or create more of them under certain situations. But in other ways, this can also be very helpful in adapting to the environment.